Okay, so this lecture was about viruses. We talked about the general characteristics of viruses, and then we talked about specific pathogens and the diseases that they cause. So, starting off with the basics, so we know that viruses are basically genetic material, DNA or RNA, wrapped in a protein coat. Okay, we know that viruses have to replicate inside of living cells. Okay, so viruses are non-living things; they are not cellular. They need a living cell to replicate, so they cannot replicate outside of a cell, and they cannot replicate inside of a dead cell. Um, and then down here at the bottom, we talked about host range, and I kind of modified this a bit in class. Host range, just basically the organisms that a virus can infect. Okay, for example, influenza A can infect a number of animals, such as humans, birds, other mammals. And then I talked about the term tropism. Tropism, these are the specific tissues that the virus will infect or have a preference to. Using the same example with influenza, again, the host range, humans, birds, swine, all of that. But the tropism is cells of the respiratory tract. Okay, so there's a difference. Okay, this is just the slide showing how much smaller viruses are than bacteria and then our own cells. So viral structure, the term virion just refers to a virus that's outside of a cell. Okay. As I said earlier, the genetic material can be DNA or RNA, but it can also be single-stranded or double-stranded. So you can really have a number of variations of viruses in terms of their genetic material. So the protein code is called a capsid, and that capsid is composed of these subunits known as capsomeres. Okay, so the capsid is the whole coat, and then the components of the coat are called capsomeres, which are just protein subunits. Now, this capsid might be surrounded by an envelope, which is really just a portion of the membrane of the host cell, or it may be naked. So if it has an envelope, we say it's enveloped. If it's naked, it's non-enveloped. Okay. Now the envelope viruses typically will have what are called spikes, which are just glycoproteins, but not all envelope viruses have spikes. Okay, then we talked about morphology of viruses. So we have the helical viruses, which are basically these long helical-shaped viruses with the genetic material inside of that helix. The polyhedral viruses, very, very common as far as human pathogens are concerned, and these are just viruses that have a capsid with many sides. The envelope viruses, so again, these are just ones that have an envelope, and these could be helical or polyhedral in terms of their capsids. And the complex viruses, these have components of polyhedral capsids as well as helical capsids. And these are things like bacteriophages and some human viruses like pox viruses. So, bacteriophages, remember, these are viruses that infect bacterial cells. So, you could say that they have tropism for bacterial cells. You could also say that's their host range, since bacterial cells are only one cell. So, here's just a picture showing some of these different morphologies. So, we have these helical types, okay? We have the polyhedral types, which have these many sides, and then we have the complex viruses, where they have components that are polyhedral and components that are helical. So, the whole goal of a virus is to get inside of a cell, to replicate themselves, and then spread to new cells. So, the basic steps that have to occur in order for that to happen are first attachment. So, viruses have to attach to its cell first. So, it has to attach to the cell. Once it attaches, it's going to enter the cell. And it might do that by receptor-mediated endocytosis, which is just endocytosis that's mediated by complementary receptors. Okay. For envelope viruses, though, sometimes just fusion of the envelope with the cell membrane is enough to allow entry into the cell. Okay. So first we have attachment, then we have entry, and then once viruses are inside of the cell, once they have entered, they uncoat. And uncoating is just when the nucleic acid, that DNA or RNA, separates from the capsid. So they literally take their coat off, okay? And then they will use the cell's machinery to replicate themselves and then be released and spread to other cells, okay? So again, these are just the general concepts you should be familiar with. First, we have attachment. Then we have entry. Okay, we enter into the cell. Then we have uncoating. Uncoating is when that capsid breaks apart and releases the genetic material, okay? All right, then we talked about specific pathogens, starting with the herpes virus family, or the herpes viridae family. Starting with herpes simplex 1 and 2, these are going to cause things like cold sores, genital herpes, herpetic whitlows, which are just basically herpetic lesions on the fingers, gladiatorum, which is a more widespread herpes infection of the skin, herpetic keratitis, which is an infection of the eye, which can be transmitted from fingers. So if you're touching a cold sore or any sort of herpetic sore and you touch your eyes, you can spread that to your eyes and can cause blindness. Okay, those are really the big things, but also meningitis, encephalitis, and some more severe infections also. But these 3 points up here, these are kind of the big ones. Okay, we talked about varicella zoster, which causes chickenpox and also shingles. Epstein-Barr virus. This is the most common cause of infectious mononucleosis. Epstein-Barr is also considered an oncovirus because it's associated with certain cancers. So an oncovirus is a virus that can cause cancer. It has the potential to elicit changes within a cell to make it cancerous. Okay, and it's associated with a couple of cancers like Burkitt's lymphoma, nasopharyngeal carcinoma. But the big one, infectious mononucleosis. Okay, then we talked about CMV, which is cytomegalovirus. Very, very common virus. Can cause mono, and it's also the number one cause of mild mental retardation of newborns. Okay, so it's really important that expectant mothers are aware of CMV. Okay, human herpes virus 6 and 7. These cause what's known as roseola or exanthum subitum, which is common in infants and toddlers. And basically, you'll have this rash that lasts a day or two, as well as a pretty high fever. And then human herpes virus 8, it's an oncovirus, which you see in Kaposi sarcoma, which is associated with AIDS. All right, then we talked about human papillomavirus. So human papillomavirus will cause your typical skin warts, will cause papillomas in the mouth and also the throat, will cause genital warts, and certain strains are also associated with cervical dysplasia and possibly cervical cancer. Alright, so then we talked about the hepatitis viruses. Starting with hepatitis A, remember A stands for acute. So causes an acute disease. It's spread by the fecal-oral route. There is a vaccine that's available, so that's important. So there's a vaccine for hepatitis A, and there's a vaccine for hepatitis B. And hepatitis B, remember I told you for both, okay? Because most of the times it's an acute disease, but does have a chronic phase in about 10% of cases, all right? And this is spread through infected bodily fluids, so through blood, sexual contact, and also possibly from the mother to the baby at the time of birth. And like I said earlier, there is a vaccine available. Now, hepatitis C, C stands for chronic. It's spread by the same routes as hepatitis B, and there is no vaccine. Fortunately, though, the most common genotype of hepatitis C, which is genotype 1, can be treated with drugs like Harvoni and is successful most of the time. That's just a fun fact, though. All right, the influenza viruses. We talked about A, B, and C. A is the big one. This is the most common. This is the one that causes the most severe forms of disease. Remember, influenza is a respiratory infection. All right. Now, influenza A will infect humans, swine, birds, a couple other mammals. Influenza B is less common and it causes a less severe disease. Now, not to say that infection with influenza B can't be serious, because it certainly can, but overall it's less serious and less common than A. And C is the least common and causes the least severe disease, typically. Again, that's not all inclusive. And then finally, we finished with the retroviruses. Now, retroviruses are special because they are RNA viruses, but they have an enzyme known as reverse transcriptase. So what they can do is they can reverse transcribe their RNA to DNA. And of course, we know that transcription is DNA to RNA, and then translation is RNA to protein. But these guys have an enzyme called reverse transcriptase that can take RNA and make DNA. And when they do that, when that's turned into DNA, that DNA can incorporate into our own DNA. And that can cause a lot of problems. And it's kind of how the virus can hang out and hide from the immune system. So the big retrovirus, there's actually another important retrovirus that causes human disease, specifically cancer. But we just talked about HIV, human immunodeficiency virus. This is, of course, the causative agent of AIDS, which is acquired immune deficiency syndrome. So the big problem here is that HIV infects T cells, specifically CD4 positive helper T cells. And when you kind of take out the T cells, the helper T cells, you really take out a lot of adaptive immunity, because we know that those CD4 positive T cells are needed to activate B cells and activate macrophages, and they're so important. So that's the big problem here. So without treatment, survival time after initial infection is about 9 to 11 years, and then antiretroviral therapies can allow near normal survival. But again, treatment's very expensive, and there are a number of other roadblocks when it comes to treatment. Okay? So as always, if you have any questions, please let me know.